platypus.
Welcome to Platinum, where future top-notchers are made. I'm Elise.
And I'm Joshua, and today we'll explore membrane proteins. Now, come on and join us as we make our journey to top 1.
These are our learning outcomes. By the end of the session, you'll be able to define, differentiate, and identify integral and peripheral membrane proteins.
Picture a cell trying to survive in a chaotic environment. It needs to import food, export waste, then receive signals, all without letting harmful substances slip through. How does it manage? Through specialized work of membrane proteins.
Before we begin, let's review the lipid bilayer. It consists of 2 layers of phospholipids with hydrophilic heads facing outward and hydrophobic tails inward, creating a hydrophobic core. This forms a stable, selectively permeable barrier. This is the fluid mosaic model, which describes the membrane as a dynamic and fluid structure. While lipids are the fundamental structural elements, proteins are responsible for carrying out specific membrane functions. But what exactly are membrane proteins? Well, it is a protein molecule that is attached to or associated with the membrane of a cell or an organelle. Membrane proteins catalyze chemical reactions, mediate the flow of nutrients and wastes, and relay information about the extracellular environment to intracellular components. They can be grouped based on how they associate with the membrane. We have A, integral, and B, peripheral. Simply, integral membrane proteins are embedded within the hydrophobic core, often spanning the membrane, while peripheral proteins associate loosely with the membrane surface or with integral proteins. We will differentiate the two based on their number 1, definition, number 2, structure, number 3, location, and number 4, function. Let's start with integral proteins.
What are integral proteins? Integral membrane proteins, also called intrinsic proteins, are permanently embedded within the lipid bilayer. Some types include transmembrane proteins, which spans the entire plasma membrane, and integral monotopic proteins, which are permanently attached to the membrane from only one side. Moving on to the structure, they contain hydrophobic regions that interact tightly with the lipid tails inside the membrane. Integral proteins are hard to remove due to their strong hydrophobic interactions. Detergents or organic solvents weaken these interactions, allowing the proteins to precipitate. Integral proteins are amphiphilic. They contain both hydrophobic and hydrophilic regions. The parts of the protein that are inside the membrane's nonpolar core have mostly hydrophobic, which are water repelling, and while the parts that stick out to the watery surroundings have mostly polar, which are water attracting amino acids. Transmembrane domains use alpha helices, which are the most common, or beta barrels to span the membrane. Alpha helices hide polar parts inside, exposing hydrophobic parts of the membrane. Beta barrels, on the other hand, form channels, especially in the outer membranes of the bacteria, mitochondria, and chloroplast. Types of transmembrane proteins include single pass, which are type 1 or 2, which cross the membrane once with different orientations. And the second one is multipass, which is type 3 or type 4, which cross the membrane multiple times, forming complex channels or transports. And lastly, lipid anchored, which are type 5 and type 6, which are covalently linked to lipids for additional membrane association. Integral proteins are found within the hydrophobic core of the membrane, either partially or fully spanning the bilayer. Next is function. Functionally, integral proteins serves as channels and transporters that allow ions and molecules to cross the membrane. Examples of these are ion channels, which form pores for ions to pass. Next is membrane-bound ATPases, which span the membrane to pump ions using ATP. Examples of these are sodium and potassium ATPases. Next is SLC transporters, which move solutes like sugars and amino acids across membranes. And lastly, aquaporins, which are water channels, which forms membrane channels to regulate water flow. Next, they also serve as receptors that detect signals from outside the cell. Examples are G protein-coupled receptors or GPCRs, which respond to hormones and neurotransmitters. Next is insulin receptors, which trigger metabolic responses upon binding insulin. And lastly, cell adhesion molecules, which function in signaling and cell communication. And lastly, they also serve as enzymes that catalyze reaction at the membrane surface. A key example, which was previously discussed, is membrane-bound ATPases. Membrane-bound ATPases not only transport ions but also hydrolyze ATP to drive active transport. Now, it's time for a checkpoint to really see if you're listening to the lecture. Pause the video if you wish to participate. So the question is, a mutation replaces several hydrophobic amino acids in the transmembrane domain of an integral protein with polar ones. What is the most likely consequence? And the answer is C. Heart this video if you got the correct answer.
Now let's move on to peripheral proteins. Peripheral membrane proteins, or extrinsic proteins, are loosely and temporarily attached to the membrane surface, either via interactions with integral proteins or with polar lipid head groups. Amphitrophic proteins are a subset of peripheral proteins which reversibly associate with membranes using amphipathic helices. They float freely in the cell but bind to membranes when needed. Their binding is controlled by the lipid type, chemical signals like phosphorylation, or changes in the membrane shape. Their structure. Peripheral membrane proteins are loosely attached to the membrane through protein-to-protein interactions. Mostly hydrophilic, these proteins do not penetrate the hydrophobic core and can be removed easily by high salt or extreme pH, and they are soluble in water once detached. Their attachment often relies on ionic bonds. These are the attachment mechanisms of peripheral proteins. We have electrostatic interactions with membrane surfaces, covalent attachment via lipid anchors, and amphitrophic proteins that, again, reversibly associate with membranes using amphipathic helices that can sense or induce membrane curvature. Moving on to their location, they are located on the inner or outer surface of the membrane but do not embed within the lipid bilayer. Peripheral proteins can sometimes cluster together on the membrane to create a platform for signaling or to help form vesicles during transport. These clusters sometimes even affect both layers of the membrane, helping cells coordinate complex tasks like responding to signals or recycling surface proteins. Although some integral proteins are embedded only on one side of the membrane, they differ with peripherals in their interaction with the lipid bilayer. Again, integrals have hydrophobic attachments, making them stronger and embedded, while peripherals associate indirectly through ionic or protein-to-protein interactions. Now, their functions. First, structural support. Peripheral proteins help maintain cell shape by linking the membrane to the cytoskeleton. Some examples include spection and anchoring in red blood cells, which stabilize the membrane and provide flexibility. Actin-binding proteins anchor the cytoskeleton to the plasma membrane, supporting cellular architecture. Next, cell signaling. These proteins participate in transmitting signals from the membrane to the cell's interior. Some examples include G proteins, which associate with receptors and relay signals to downstream effectors. Adapter proteins such as GRB2 link activated receptors to intracellular signaling pathways. Next, anchoring. Peripheral proteins anchor the membrane to other cellular structures or to the extracellular matrix. Some examples include ERM proteins, which connect the plasma membrane to the actin cytoskeleton. We also have focal adhesion proteins, which help anchor cells to the matrix, facilitating cell adhesion and migration. Now it's time for a checkpoint. If you wish to participate, please pause the video. What type of interaction primarily holds peripheral proteins to the membrane? And the correct answer is C. Electrostatic and hydrogen bonds with polar head groups.
Moving on to the comparison of integral and peripheral membrane proteins, first let's tackle their structural differences. For the differences of their association, integral proteins are permanently embedded in the bilayer, and peripheral proteins on the other hand are loosely attached to the surface. Next is their lipid interaction. Integrals use hydrophobic interactions with lipid tails, and peripherals rely on electrostatic bonds with head groups or proteins. For their removal, integrals need detergents, while peripherals can be removed with pH or salt changes. For the structure, integrals often have alpha helices or beta barrels and are amplified. On the other hand, peripherals lack transmembrane parts and are water-soluble. Now, for their transmembrane domains, integrals may span the membrane once or many times, while peripherals do not span the membrane. And lastly, for their stability, integrals are stable and embedded, while peripherals on the other hand are dynamic and reversible.
Now it's time for their functional differences. First, their primary roles. Integral proteins handle transport, receive signals, catalyze membrane reactions, and support cell adhesion. While peripheral proteins focus on structure, signal transduction, anchoring, and enzymatic regulation. In terms of transport, integral proteins form channels and carriers, while peripheral proteins don't transport molecules directly. Next, signal reception. Integral proteins act as receptors for external signals, while peripheral proteins relay those signals inside the cell. Enzymatic activity. Integral proteins catalyze reactions at the membrane surface, while peripheral proteins may help in lipid metabolism or signaling like phospholipases. Structurally, integral proteins maintain membrane integrity and help with adhesion. Peripheral proteins connect the membrane to the cytoskeleton, supporting shape and stability. Cytoskeleton interaction. While integral proteins link indirectly via adapters, peripheral proteins bind directly to cytoskeletal elements or extracellular structure. Finally, dynamics and binding. Integral proteins are stable and embedded long-term, while peripheral proteins bind reversibly, making them more dynamic in function. Now it's time for a checkpoint. Identify if the protein is integral or peripheral based on its characteristics. If you wish to participate, please pause the video. And the correct answers are B, peripheral, and A, integral.
Moving on to their biological importance. Membrane proteins are vital for many cellular processes. Integral proteins control the selective transport of ions and molecules across the membrane. It also acts as receptors to detect external signals and catalyze enzymatic reactions essential for cell metabolism. Peripheral proteins support the membrane structure by linking it to the cytoskeleton and play key roles in their intracellular signaling and membrane organization. Together, these proteins enable cells to communicate, maintain shape, regulate their internal environment, and respond to external changes.
Moving on to the clinical applications of membrane proteins. Membrane proteins are major targets in medicine due to their accessibility and functional importance. Integral membrane proteins, especially GPCRs, ion channels, and transporters, are targeted by many drugs treating hypertension, cancer, diabetes, neurological disorders, and more. Mutations or dysfunction in membrane proteins can cause diseases such as cystic fibrosis, a defective chloride channel, cardiac arrhythmias, ion channelopathies, and cancers, altered receptor signaling. Membrane proteins often serve as biomarkers for diagnosis and prognosis, and are targets for vaccine development due to their extracellular exposure. Understanding membrane protein structure and function is therefore essential for advancing diagnostics and personalized medicine. For our final checkpoint, classify if the labeled protein in the diagram is an integral or peripheral protein. If you wish to participate, please pause the video. And the answers are number 1 integral, number 2 peripheral, number 3 integral, and number 4 peripheral. And this concludes our lecture on membrane proteins. We hope you are now able to define, differentiate, and identify integral and peripheral proteins. Thank you so much for listening, and remember, strive, excel, succeed. This is Platinum, where future top-notchers are made. See you on the next review.